So let's start with glycolysis. Just a minute. Yeah, let's start with the glycolysis. Okay, so glycolysis is composed of two words, glyco plus lysis. Okay, so glycolysis, that means glyco means sugar and lysis means to break down the sugar. So in glycolysis, what we do, we utilize the glucose, which is our carbon source, and break it down to get the energy from this particular molecule. Okay. So glycolysis, we start with glucose, which is a 6 carbon molecule. It is a 6 carbon molecule. Okay, and after the entire glycolysis process, this 6 carbon molecule will break down in two 3 carbon molecules. That is known as pyruvate. Okay, so during this, from this 6 carbon molecule to 3 carbon molecule conversion, this breakdown produces energy, and that energy is produced in the forms of ATP and NADH. Okay, so this is the glycolysis process. So if I'll write the entire reaction, so net reaction is glucose plus NAD plus, and this came from this 2 ATP, which will generate 2 pyruvate molecules, 3 carbon molecules, and 2 NADH. ATP. So conversion of NAD plus to NADH. Is it reduction or oxidation? What is happening here? It is a reduction reaction which is taking place here. So this is the net reaction. So at the end of our glycolysis, we'll be having 2 molecules of pyruvate, 2 NADH and 2 ATP, which will work as an energy currency for the cell. These ATP and NADH, which will be further break down into ATPs. Okay. So this is the net reaction. So if we'll start about the glycolysis, so glycolysis is a total 10-step reaction, right? Everybody knows. 10-step reaction. So if we have to describe glycolysis, we can divide it into 2 parts. One will be our investment phase, we can say. Investment phase. Another, which is our first phase. And the second phase, which we can say our payoff phase. Investment and payoff. So from step 1 to step 5, we have our investment phase. So investment phase is nothing, it is like energy consuming phase. So where in the beginning of the glycolysis, you have to provide energy so that the reaction can follow up, and then in the second part of the glycolysis, which is called as payoff phase, where the energy is being produced. So here energy is being consumed, and here the energy is being produced, which include from step 6 to step 10, like that we can say. Okay, So during the first step, it consumes total 2 ATP, and at the end of the during this payoff phase, it generates 4 ATP and 2 NADH. Okay, so if I need to say net production of energy, then how many ATP will be produced? Yes, will be 2 ATP and 2 NADH. So this is the net energy production from the glycolysis. Okay. So we start the glycolysis from the first step is glucose. So glucose enters inside the cell, and then this glucose is being converted to glucose 6 phosphate. Okay. Structure wise, if I have to say, so this is the glucose structure. Sorry. So, this is the first, second, third, fourth, fifth, and sixth carbon. This is glucose. So it is being converted, or it is being phosphorylated at sixth position, which is the sixth carbon, and it is being generated. Now this sixth carbon is phosphorylated. Okay. And this is our glucose 6 phosphate, and this is carried out by enzyme called hexokinase, which utilizes one ATP, converts it to ADP, and this one phosphate group of ATP is joined at the sixth carbon position in the glucose structure and makes it to glucose 6 phosphate. Okay. So this step is very, very important because cell takes up the glucose. So this glucose can cross the membrane, can enter inside our cell, and when this glucose is converted to glucose 6 phosphate, now this becomes a charged molecule, and this charged molecule cannot pass the membrane. So once the glucose enters inside the cell, our cell tries to trap the glucose inside our body or inside our tissues and cells. So that is why this step is very important, and this charged glucose cannot go back across the cell membrane to come out of the cells. Okay, so this is the trapping of the glucose using the hexokinase enzyme. So this hexokinase enzyme is present all our cells, all of our bodies. So there is one isomer of this particular enzyme, that is glucokinase. So these both are isozymes, okay, perform the same function. So glucokinase is nothing but hexokinase D. So this is this hexokinase D. It is a similar form of glucokinase, which is called as hexokinase D. It's a form of glucokinase. So this glucokinase is present in our liver cells where we store the food. Okay, so it is present majorly in hepatocytes, trapping the glucose inside our body. And if we compare these two, hexokinase and glucokinase, so this glucokinase have lower affinity towards the substrate. That means it has high Km value, and this one has the low Km value. That means high affinity towards its substrate. Okay, so this could be a question in some of the examination. That is why I mentioned it here. So once the glucose is trapped in form of glucose 6-phosphate, the next enzyme which works on it is phosphoglucoisomerase, which converts this glucose 6-phosphate to fructose 6-phosphate. Fructose 6-phosphate. So this is our fructose 6-phosphate, just isomerization of this particular glucose 6-phosphate to fructose 6-phosphate. Okay. And then this fructose 6-phosphate again gets phosphorylated. Again, one more ATP is consumed at this step. Okay, so this is the first step, second step, third step. So at first step and third step, at both the steps, ATP is being consumed. Okay, ATP is consumed, converted to ATP. And then this fructose 6-phosphate is converted to fructose 1,6-phosphate. So this is fructose 1,6 this phosphate. So this structure, and then this, there is a cleavage in this reaction which is performed by aldolase enzyme, and this breaks the chain, this cyclic, and make two structure, one which is called as glyceraldehyde 3-phosphate and another which is dihydroxy acetone phosphate. You will get two structure, dihydroxy acetone phosphate and glyceraldehyde 3-phosphate. So it is divided into two. Okay. Three phosphate. This is glyceraldehyde 3-phosphate, and we'll have dihydroxy acetone phosphate as well. So these two structures will get, which are interconvertible with each other. So here the enzyme which works is the triose phosphate isomerases. So that converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So now we started from one glucose molecule, and after these many steps, the 5 steps of the reaction, we'll have two glyceraldehyde 3-phosphate from one molecule of glucose. Okay. From one glucose, we have glyceraldehyde 3-phosphate and glyceraldehyde 3-phosphate, two molecules of glyceraldehyde 3-phosphate. And now they both will go with several breakdown in the similar way. And here we have consumed two ATPs to generate two molecules of glyceraldehyde 3-phosphate. So this is our step 1 to step 5. And from here, which is our payoff phase, from step 6 to step 10, here in these phases, it generates the energy. Okay. So here also we'll see the two enzymes. One is the hexokinase enzyme and this one, phosphofructokinase enzyme. Here works here. phosphofructokinase. So these two steps are the ATP consuming steps, and if we'll see, these two steps are the irreversible reaction steps. Other steps can be like this, okay, but these two steps are the irreversible steps, okay. So from here onwards, glycerol glyceraldehyde 3 phosphate. So this particular component, it now gets converted. Glyceraldehyde 3 phosphate, it now gets converted to 1,3 bisphosphoglycerate. The structure was something like this. This is our glyceraldehyde 3 phosphate, and it gets converted to 1,3 bisphosphoglycerate with the help of NAD plus. Okay. So NAD plus gets reduced to NADH and oxidize this particular aldehyde to the carboxylic acid. So it is a two-step reaction. So in first step, what happens? First, the oxidation of this aldehyde takes place to make carboxylic acid, and then in the second step, the inorganic phosphate, the orthophosphate, joins at that particular position So this is 1,3 bisphosphoglycerate. So this structure gets formed. Okay. So now this particular phosphate is a very high energy phosphate. Okay. And this formation is this particular component gives rise to the production of ATP. So ADP takes the phosphate group of this particular high energy bond from here, and it converts it into the ATP. Okay, and thus we get left with this structure. Just this 3-phosphoglycerate. Okay, and here we get one ATP, and this ATP we are getting after phosphorylating this ADP, and this phosphorylation we are doing using the substrate of the reaction. So this particular reaction is called substrate level phosphorylation. Substrate level phosphorylation. So this is one substrate level phosphorylation where we are phosphorylating ADP to produce ATP, and this phosphorylation is happening with the help of our particular substrate in the reaction. And then we are getting 3-phosphoglycerate, which now rearranges itself and forms 2-phosphoglycerate. H2. Two phosphoglycerate, and this step from here it gets converted to phosphoenol pyruvate with the help of enzyme called enolase. So here the enzyme which plays the role is phosphoglycerate kinase. Okay. Phosphoglycerate kinase. That is the enzyme which is involved in substrate level phosphorylation. Okay. Then we get 3-phosphoglycerate, which gets converted to 2-phosphoglycerate using mutase enzyme, and this 2-phosphoglycerate further gets converted to phosphoenol pyruvate, which have this structure. Here the water molecule is being produced. Okay, and the water molecule being produced, and this we get our structure, which is called phosphoenol pyruvate, which is having very high energy enol phosphate group, and that phosphate group yields another ATP via substrate level phosphorylation. Another ATP is produced at this step, and we get our pyruvate structure. I'm drawing here. So we get so this is our pyruvate which we are getting, and here we are getting one ATP. So this is also another substrate level phosphorylation which is happening from this substrate phosphoenol pyruvate, and the enzyme which is involved at this substrate level phosphorylation is named as pyruvate kinase. Pyruvate kinase. Okay, so in this step 6 to 10 we have the two substrate level phosphorylation, and the enzymes involved are labeled as phosphoglycerate kinase and pyruvate kinase, and they produce. So here in this particular step we get one NADH, one ATP, another ATP, so total 2 ATP and one NADH from one glyceraldehyde 3-phosphate. So similarly, if we have two molecules, we'll have 2 NADH and 4 ATPs. But as 2 ATPs are already consumed in the beginning of the reaction, these 2 ATPs, so we'll see. So if we'll see, we'll have net gain of 2 ATP and 2 NADH. Okay, so this is our whole glycolysis cycle which is taking place in the cytoplasm. At the end we are getting the pyruvate, and this pyruvate further goes inside the mitochondrial matrix, and there it performs the TCA cycle. First it undergoes oxidative decarboxylation, then TCA, and then it goes to oxidative phosphorylation step to produce ATP. Okay, so we'll see our first question which appeared in GATE 2019 exam. So it's question number 3. It says the example of substrate level phosphorylation in glycolysis is. Okay, so 4 options are given. I'll read and you tell me whether it is correct or not. So first option is conversion of glucose to glucose 6-phosphate. Is it substrate level phosphorylation? This one is not. Then second one is conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Is it? Here also one more phosphate group is attached to the further or our product of the reaction. So here ADP is not getting phosphorylated. So the substrate phosphorylation we are saying there, where we are phosphorylating ADP to produce ATP, and this particular phosphate group is coming from our particular substrate. Okay, so third option is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. Is it? Yeah. So here 2 phosphate groups are there. So we have a structure. Our substrate, and here the phosphate group is being added to ADP to produce ATP, and we are getting, so this is 1,3 bisphosphoglycerate, and we are getting 3 phosphoglycerate after the end of the reaction. So this is our 3 phosphoglycerate. This is our 1,3 bisphosphoglycerate. So this is the correct option. Fourth option is conversion of dihydroxyacetylene phosphate to glyceraldehyde 3-phosphate. Now this is also interconversion. They both have the same structure and number of atoms in it. So this is just interconversion using the enzyme isomerase. Okay, so the correct option would be third option. This is my NPTEL reference where you can read in detail about glycolysis. Thank you.